Welcome back. All right, now that we've talked about how it is that we know that DNA is the chemical component of heredity, we need to actually get into the molecular genetics aspect of the course. And so we need to talk about first DNA replication, the next steps. Once we knew it was DNA, that is how we transmit our or transfer our traits to our offspring, then the question was, well, okay, how does DNA replicate? Because Watson and Crick put out that paper in which they said that it was immediately apparent how it could be replicated, but in fact, it wasn't. So there were multiple theories as to how DNA is replicated. All of them fall within what Watson and Crick proposed, but exactly how that occurs at a molecular level is what we'll look at. And then that'll set us up for the central dogma of molecular biology. And so then from there, what we'll do is we'll talk about transcription, RNA processing, translation, and essentially what we're working our way up to is understanding gene expression so that we can learn how gene expression is going to be regulated. So let's start with how DNA is replicated because all cells must replicate their DNA in order to survive. Now let's start with the slide. The whole thing pops up because, of course, you've seen this slide before. It was at the beginning of our last lecture when we first started talking about, the chemical component of heredity. And so the thing that we've already covered now is that, remember how we said even before we knew what it was that was how we carry on or how we pass on our traits, biologists recognized genetic material must do certain things. Well, contain complex information in a stable fashion is essentially what we'll talk about when we talk about gene expression, so transcription, translation. What we're going to focus on right now is, and actually to a certain extent, we'll also talk about how it encodes the phenotype when we talk about gene expression. So those two bullet points are coming up. But right now, what we can focus on is basically how DNA is capable of replicating faithfully and with high fidelity. So that's why we're going to talk about DNA synthesis, because it's important to know how DNA replication comes about. It's absolutely essential to life on this planet. And then on top of that, it's also going to be one more experiment that you're going to be responsible for. So in addition to the 3 experiments we've talked about previously, so Hershey-Chase, actually let me go in order. So Fred Griffith's experiment, then Avery McLeod-McCarty, then Hershey-Chase. You've got one more that you're going to be responsible for, and that one is going to be the Michelson and Stahl, or some people say Michelson and Stahl experiment. So let's start with how accurate this must be. This process must be absolutely accurate. So errors arise whenever information is copied. Remember we talked about, you're going to have, I don't know if we mentioned it, but you're going to have one mistake per billion nucleotides, which sounds really awesome. Sounds like, wow, that's like 1 in a billion. That's amazing. But you have 3.2 billion base pairs of DNA twice in every cell. So that's 6.4. So let's round down and say 6, mistakes for every time you replicate the DNA in a cell. And then, of course, you have trillions of body cells. So let's say 100 trillion body cells. Well, now you have 600 trillion mistakes that you make every time you turn over your cells. And we are rapidly replicating the DNA in our cells and making new cells all the time. So that's actually a fairly substantial amount of error, which is good from a species point of view because it ensures that we will always have the ability to mutate our DNA and therefore evolve. But from a personal point of view, obviously, it's not the best because that's how we age. That's what leads to certain genetic diseases, sickness, that sort of thing. So errors always are going to arise whenever information is copied. The more times it's copied, the greater number of errors. And genetic information has to be accurately copied every single time a cell divides, and it has to be copied at just an astoundingly fast speed. Because let's think about this. I just mentioned 6.4 billion base pairs of DNA. You need to copy that. And that can't take up the entire life of the cell. In fact, it only takes up just S phase of interphase, if you recall from when we went over the cell cycle. So here's E. coli. You've seen this image before, 4.6 million base pairs, but doubles in only 20 minutes. So, and you don't take the entire 20 minutes to replicate that 4.6 million base pairs of DNA. So it's only a fraction of that 20 minutes in which you replicate that 4.6 million base pairs of DNA in E. coli. So DNA polymerases are the enzymes that actually do that. They're the ones that make the new DNA from the old DNA. So they synthesize DNA. And there are many DNA polymerases, both in prokaryotic life and eukaryotic life. There's not just one. So you can see some DNA polymerases synthesize DNA at over 1,000 nucleotides per second. So think about that. These are 1,000 characters of code per second. Imagine trying to type 1,000, just not even words, just letters per second, right? I mean, it's unlikely that you'll be able to do that. And so these polymerases are doing so. They're zipping along the template DNA, and they are laying down new DNA at 1,000 nucleotides per second with less than one error per billion nucleotides. So 1 in a billion mistakes. I mean, mistakes are 1 in a billion. Let me try that again. And so that sounds awesome. And so to achieve this speed and accuracy, polymerases require a smooth track. So you need mutation-free DNA. So you need the DNA to be opened up. You need it to be unwound. You need a whole bunch of other factors to come into play. We'll talk about helicases and all these other things. But you need all these factors to kind of lay down a smooth track for the DNA. And then it actually has to start off of a little RNA primer. But when it does, it then zips along at 1,000 nucleotides per second. Now, there are other DNA polymerases that aren't as fast. They're slower, less efficient translesion polymerases. And those are there to actually fix the obstructions, so to fix mutations. So we have, imagine just typing, right? So let's say you're a DNA polymerase and you're typing pretty fast, and you make a mistake. Well, some DNA polymerases have an error proofing mechanism where they basically proofread their work. So you're typing, and you're typing in some word processing program. You see that little red like underline underneath saying, oh, you misspelled a word. Well, then if you catch it, you could say, oh, I see that, and you can backspace, and then you can basically retype that word. DNA polymerase is capable of doing that, the fast one. And then on top of that, then after you're done, you should use the spell checking mechanism or function in whatever word processing program you're using. And so when you do that, now you're using these slower translesion DNA polymerases that are going and looking for misspellings or whatever it may be. And then they find them and then you can correct them. And so obviously we're not going to correct everything, but all of this contributes to that fidelity, that making it more and more accurate. So when errors are made, most are corrected by DNA repair mechanisms. The proofreading of the DNA polymerase is one thing while it's being synthesized, but then after the fact, you have DNA repair mechanisms. So here are some DNA polymerases from eukaryotic cells. You can see there's basically what you want to remember is you don't have to remember all of these and their functions individually. I will ask that you remember that the eukaryotic DNA polymerases have the lowercase Greek letters, right? So alpha, beta, gamma, delta, epsilon, you get it. And so they have, so these are DNA polymerases. So I would argue that one of the dumbest columns in the entire book in a table is this one. So all of these have DNA polymerase activity, 5' to 3' DNA polymerase activity. I say that's, kind of silly to include that because it's a polymerase, and so it is going to polymerize DNA, and that's why it's classified as a DNA polymerase. And one thing that I will mention is that you only construct DNA in a 5' to 3' fashion. So it's literally saying, here's the DNA polymerase, and does it do exactly everything it's supposed to be defined to do? Yes. Let's make a whole column for that says yes. Now, over here, this is actually a column worth having. This is 3' to 5' exonuclease activity. This is cutting out stuff. This is the backspace button. So does DNA polymerase alpha have a backspace? No. Does DNA polymerase gamma have backspace? Yep. So that one can basically, you could back up and lay down new DNA where after it makes a mistake. And so you can see their function. There's quite a few functions. They're all involved in some form of DNA synthesis or DNA repair, which involves DNA synthesis. And so those are the eukaryotic DNA polymerases. Now, certain diseases exist where you have defects in the genes encoding DNA synthesis or repair enzymes. So BRCA1, BRCA2 are very famous genes that are mutated in people that have a higher propensity for breast and ovarian cancer because they are DNA repair mechanism enzymes. And so when they're mutated, they don't repair, mutations that accumulate, and so that can lead to a higher risk of cancer. Similarly, here is an example of a little guy who had, xeroderma pigmentosum. And xeroderma pigmentosum is basically you lack so our skin, when you walk from here to your car, let's say you walk outside and you get hit by the sun for a little bit, you're getting hit with some UV. And that UV causes what are called thymine dimers. So this is a T base, and it's supposed to be hydrogen bonding here with A's, but it's not. Actually, over here you can see what it's supposed to look like. So here's A, here's T. Oh, let me try that again. Here's T, here's A. And so 2 hydrogen bonds between them. But here you don't see those hydrogen bonds because the UV light caused adjacent thymines to covalently bond to one another. It's called a thymine dimer. That's a mutation. And we have DNA repair mechanisms that can fix that. Unfortunately, if you have xeroderma pigmentosum, you do not. And so you can see that even at a young age, he's got tremendous pock marks and freckling, and he's going to develop, if not already, skin cancer. It's unfortunate. He cannot be exposed to any UV light without DNA damage, permanent DNA damage in his case. So all DNA replication turns out to be semi-conservative. What that means is, and you can see that in the image here, that the new DNA molecules are going to be half old, half new. Right? So it's not conservative where we open up the old DNA molecule, we make our new ones, we take our 2 new ones, we put them together, and then we take our 2 old ones, put them together. That'd be conservative. That's not it. And it's not dispersive, which is, I'll show you all 3 of these mechanisms because, or we'll at least talk about them because they were the 3 prevailing theories as to how we replicate our DNA. And dispersive is literally the DNA just gets broken up into a bunch of pieces. You replicate each fragment and then you put them all back together. So as you can imagine, that is like if you were to break something up into puzzle pieces and then replicate and then try and put it all back together, that's a tremendous amount of extra work. So that's not very simple. Whereas conservative replication is you take apart the old DNA molecule, replicate, then put the new ones together and put the old ones together. There's still an extra step there. And if we follow Occam's razor, where the simplest explanation is usually true, well, then the simplest mechanism that, millions and millions of years of evolution could have come up with is semi-conservative replication, which is you take the old strand, you open it up, you make new strands on the 2 old strands, and you're done. And so it's the fewest number of steps. So think of this as a zipper, right? As you're unzipping it, you make new strands on the old strands, and then when you get to the end, you have 2 double-stranded DNA molecules, identical in nature. And so that would be the simplest mechanism. But we didn't know whether it was dispersive or conservative or semi-conservative. So each DNA strand is in a double-stranded molecule is complementary to one another, and each can serve as a template for a new strand. So 2 DNA molecules can be built on a pair of templates, and they'll be identical. These DNA molecules are going to be identical to one another. This sort of DNA replication, the one that's shown in this image, is semi-conservative, as I've said earlier. So here it is in a bullet point. Semi-conservative means each daughter strand is composed of 1 new strand and 1 old strand. Half old, half new, as I call it. Now, but for a while, 3 alternative models were proposed for DNA replication. So here's the 3 models that I mentioned, verbally. Here it is in an image. So conservative replication, you can see you open up your DNA molecule, you make you take you use each strand as a template strand, and then you put your old DNA back together, you put your new DNA together, and so you can see that you conserved the original DNA molecule, and that gets conserved through successive rounds of replication. Alternatively, we have dispersive, and it's hard to see in this image, but it's literally just imagine chopping up this DNA molecule, then making a whole bunch of little half old, half new fragments, then just putting them back together in the correct order, regardless of whether it's new or old. And so you could see that just continues. That would be dispersive. And then what I did show you on the previous image is semi-conservative, half old, half new. So open up the blue, and then you can see the first round is going to be half blue, half red, half blue, half red. So half old, half new. And then this is going to open up, and we'll have blue and red as templates. And then the blue is going to end up being half old, half new again. The red is going to get a new red made on it, and so it's going to be completely new at this point. And so that's semi-conservative replication, where you're always, after the first round of replication, you're always going to have 2 that are half old, half new, and then all the rest are going to be all brand new DNA molecules, newly synthesized double-stranded DNA molecules. Now, I mentioned a moment ago, Meselson and Stahl, or Meselson and Stahl, depending on how you want to pronounce it, provided they actually performed an experiment that provided the necessary evidence to conclude which one of these three forms of replication was the correct form of replication. And so they were able to prove replication is semi-conservative. Here's what they did. So they started with a way they wanted to be able to follow the old DNA, the original DNA. So what they did was they grew up cells in N15 containing medium. So nitrogen is its atomic mass. If you look on the periodic table, you'll see it's 14.01. So it's basically N14 is the most common isotope that we find in nature. So regular nitrogen is N14. Heavy nitrogen would be the N15 isotope. And so they grew cells in N15 containing medium so that all of the DNA that they made had N15, thereby making that DNA, because it had a tremendous amount of nitrogenous bases in it, heavy. So they made heavy DNA containing cells. Then what they did was they spun this down. So they had a pellet of cells. And they took before they spin it down, they take a little aliquot of it and they do density gradient centrifugation. I'll talk about that in a second. And then what they did was they basically spun it down, poured off the medium, and then resuspended in regular medium. So this is now regular old nitrogen containing medium. So there is any new DNA that's going to be made from here on out after this first step, this transfer to N14 medium. Any DNA that's going to be made from here on out is not going to be heavy DNA. So we can follow the original DNA molecules. And so, like I said, they took a little aliquot of this. And so it's a funny story. I had no idea because, being in science my entire life, I had no idea that aliquot is not a normal everyday term. And my wife laughed at me tremendously when I told my kids to go get little aliquots of ketchup from the thing at In-N-Out Burger one time, the little cups and you go get ketchup. And so I'm like, just go get a couple aliquots of ketchup. And then the three of them just looked at me like I was crazy. And so we had this discussion. But apparently, aliquot is not everyday speech. So I am going to ask you formally to please try and incorporate aliquot into your everyday speech so that I'm not the weird one anymore. But anyway, so they took a small volume sample of this medium before transferring it, right? So that contains cells. That would be the aliquot. And then they lysed the cells open and they spun them down in a centrifuge tube containing material that, as you can see, as it gets darker, as it goes down, that represents the density. So it becomes more and more dense as it goes towards the bottom. That's the density gradient. They spin it, density gradient centrifugation. And if it's heavy, then it will reach neutral buoyancy further down in the tube while you're spinning it, and it won't travel any further because that's, basically its density will equilibrate to that section of the tube. So, as you can see, heavier things that are more dense are going to be lower in the tube. So the heavy DNA spins down pretty far down in the tube. That's our original DNA. Then we basically transfer it to medium, and then it grows for one cell cycle, right? One round of division. And so let's say we're dealing with E. coli, so we take a sample every 20 minutes. And so we have logarithmic growth, and we have new cells being made with new DNA. And And so what we have is you just continue to passage these cells in their medium. You take an aliquot every generation, and you do the same thing. You lyse the cells open, you basically spin it down, and you see where the DNA comes out. And so their experiment showed us that the very first step of their experiment, they had heavy DNA. The second round, after one round of cell division, they had hybrid DNA that was half heavy, half light. It wasn't quite as heavy as the heavy DNA. It wasn't light as the light DNA would be expected to be. And so we have, basically what appears to be, as you now know, half old, half new DNA. So that would cut its density in half, right? And then this doesn't go away. This stays for the rest of the experiment. We have a small amount of half old, half new, but we have increasing amounts of light DNA after the second round of division and on. And you can see that here. So again, you saw this on the previous slide. This is semi-conservative replication. This proves semi-conservative replication. What you see is that the original DNA is going to open up, right? So you basically separate this into two molecules, and then half old, half new, half old, half new. That's what you have here. And then from here on out, that one's always going to give you, this molecule, because it's got a blue, is always going to give you a half old, half new. This one's always going to give you a half old, half new. But the red here is going to be all new when we give it a partner. And the red here, when we replicate a new red for it, it's going to have all new. So now you can see, we see this. And then what you can't see in this image, what they did see in their experiment, is that the light DNA after this point starts to accumulate. It starts to get darker and darker because you're increasing the concentration of it with every successive cell generation. And so here you can see they're actually their original data. So this is from a 1958 paper. And so you can see it's from measles install, replication of DNA in E. coli, and it's showing semi-conservative replication. So here's that first band, right? That was really heavy. And then, you know, I know you see other things in here, but you got to remember that it's basically, you know, it's just experimental error, right? So that is stuff that didn't quite spin down all the way. Or, you know, I guess I have to go back. I could say, you know, I have to read the paper more closely. That could also be like maybe they didn't quite, you know, they already transferred. I'd have to see. And so, oh, well, here you go. Generations after N14 addition. So one way to interpret this is, okay, well, now the cells are starting to make half old, half new. Or sorry, yeah, half old, half new DNA. So that's why it's 0.3. So we're not quite all the way there. But anyway, so here's, you know, what you started with. Then by one generation, the heavy is gone. So they're showing the transition. The heavy is gone, and the half old, half new, medium sized, right, half heavy DNA is 100% present after one generation. And then you follow it, and this never goes away. It's starting to get diluted out because it's been there for a long time. So you can imagine, in order to get an exposure of the radioactivity to actually see it like this without having it blow away everything, there wasn't enough time to see the smaller concentration of this. But you can kind of see it there. But as you can see that as we go from here on out, we have more and more. So this is just 1.1, nothing really going on there. 1.5 there. This is basically 2. So now you can see that, and 2.5, 3. So you can see that, and you can clearly see that the new DNA is accumulating because it's all light DNA. So this data shows that measles and install were able to prove that DNA replication is semi-conservative. So there are several different ways in which semi-conservative replication can take place. And so they differ in the nature of the template DNA, whether we have linear chromosomes like we do in eukaryotic cells, or whether we have circular chromosomes like we do in prokaryotic cells. So in either case, whether it's prokaryotic or eukaryotic, replication is going to start at the origin of replication. And it will continue until the entire replicon has been replicated. A replicon is really just the sequence of DNA between an origin of replication and another origin replication, or an origin of replication and the end of a chromosome. So that would be a replicon. Like, for example, if you had a linear chromosome with, say, I don't know, 2 origins of replication, draw it out. Here, let's do it. So you would have, so here's your linear chromosome. And let's say we had an origin of replication there, origin of replication there. So if we have that, how many replicons do we have? We have 3, right? So we have 1, 2, 3 replicons. Common mistake. Don't say 2. If you have 2 origins of replication, you have 2 replicons. No, you have 3. You have 3 in this case. All right, so let me get rid of that. Okay, and so here we go. So bacterial chromosomes only have a single origin of replication because they're circular. Once you start and you go bidirectionally left and right away from it, you're going to come back around and connect. Eukaryotic chromosomes, on the other hand, have lots and lots of replication origins. And so in bacterial cells, or basically anything with a circular chromosome, we call that form of replication theta replication. And it's called theta replication because it actually at one point the replicating chromosome looks like the lowercase Greek letter theta. So you can see right here, this would be where we're kind of in a theta, like that right there, right? Kind of looks like a theta. And so, like I said, you just need one origin of replication, because then you can go either way. And you can see these bidirectional replication. These, replication bubbles are going to move, bidirectionally. They're going to meet. And once they meet, you're going to have two DNA molecules. And so here again, there's what theta replication, that's why it's called that. Note that the DNA unwinds at the origin, producing a replication bubble. So I used that term already, but we need to know our terms before we get into DNA replication. So there's the origin of replication, and then when you start to open it up, you make a replication bubble. And we will also talk about a replication fork here in just a second. So DNA is synthesized on two single-stranded templates at each of the two, as I said, we would talk about it, two replication forks. And I'll show you some images of replication forks here in just a second. So before we get to that, let me just say replication proceeds in opposite directions from an origin of replication. So it's called bidirectional replication. You heard me say bidirectionally. Well, that's because that's the term we use. It's going, this way as well as this way. So it's bidirectional replication that's occurring, emanating from these origins of replication. So this type of replication is accomplished from a single origin. Theta replication is accomplished from a single origin because the chromosome is small and it doesn't take too long to replicate. You can just get away with one. It's a small chromosome. It's circular, so you're basically going in opposite directions anyway. You're doing great. You're very efficient. When you have much, much longer linear chromosomes, such as what we have in eukaryotic organisms, that's when you need multiple origins of replication. So larger chromosomes have multiple origins of replication. And eukaryotic replicons can be anywhere from 20,000 to 300,000 base pairs in length. So here is a eukaryotic chromosome because it's linear. All right, we can all right off the bat, that cartoon of a eukaryotic of a chromosome, you already know it's a eukaryotic chromosome for two reasons. It's linear, and you see multiple origins of replication. So those are two clues that it is a eukaryotic chromosome. Then the thing to keep in mind is that you may not start at all of these origins of replication simultaneously, as you can see. There's a protein called licensing factor that will determine when the origin of replication is licensed, when it's allowed to actually start the process of replication. Now, there are many reasons for this, but simply put, you want to be able to finish all of the chromosomes at the same time. You don't have to start everywhere at the same time, but you need to finish at the same time so that we can move out of S phase of interphase and into G2 of interphase, you might recall from our discussion of the cell cycle. So right now, this picture is showing us what's occurring on a chromosome during S phase of interphase. And so just replicating. And so you can see, well, here's three origins of replication, 1, 2, 3, 4 different replicons. Eventually, some meet earlier than others, but eventually they all meet. And when they all meet, you now have two newly synthesized molecules of DNA. Okay, so there are three things that you have to keep in mind when DNA is being replicated. DNA replication requires three groups of materials, and they are, of course, you have to have a single-stranded DNA template. Now, it may seem like common sense. Yeah, you got to have a template. How are you going to make more DNA without knowing what to use as a template, without knowing what you're copying, right? How do you copy something if you don't have the original? But that template needs to be single-stranded, and to make it single-stranded is actually a chore. Raw materials, obviously you need to have nucleotides, so they're known as DNTPs, N being a variable for A or G or C or T, and D for deoxy. So these are your ribonucleotides, deoxyribonucleotides, triphosphates. You need other things, magnesium. We won't get into the chemistry of it, but you need a number of other things as well. And then, of course, you need enzymes. One of the, we're not going to talk about absolutely everything that's necessary because that would be very intensive, but we do need to talk about the most important aspects of DNA synthesis. And of course, we've talked about DNA polymerase, but there are other enzymes that are involved in things like unwinding the DNA. Obviously, you need to be able to read the template. You need to assemble new strands of DNA. You need to supercoil the DNA. You'll need to lay down RNA primers. You'll need to, synthesize little covalent bonds between the sugar phosphate backbone of strands of DNA so that you can finalize it, kind of weld it all together. There are all these enzymes that we'll talk about that are involved. And so in DNA synthesis, DNA polymerases and, I should say add DNTPs to the 3' hydroxyl group of the growing strand. Now, this is an important bullet point that can be summarized as this. The 3' end is the growing end. And so DNA is synthesized in a 5' to 3' fashion. So that means that the 3' end is the growing end. The 3' end is always the growing end. And so therefore, DNA polymerase synthesizes molecules, or DNA is, ribonucleotides. So it doesn't even matter if it's DNA or RNA, it's just polynucleotides are synthesized in a 5' to 3' direction. The 3' end is the growing end. So DNA is synthesized only 5' to 3' in a 5' to 3' manner. Can't stress that enough, because if you know that, then you can appreciate why there has to be all these extra steps in order to maintain that directionality. And you can see that here. So here's the original template strand. And if that is, if you show it going this way, 5' to 3', that means that we have to be making DNA 5' to 3' in the opposite direction because they're anti-parallel. And so you can see there's that 3' hydroxyl group. And so if you're familiar with the sugar, right, you may have forgotten. Remember, the base gets the 1, 2, 3 designations, and then in organic chemistry, you have to give everything an address. And so the numbers of this, the sugar, is going to get the carbons are going to get a 1', 2', 3', 4', 5' sort of designation. And so you'd have, your 1', 2', 3', 4', 5'. And so basically, that is the 3' carbon. And there's a hydroxyl group sticking off of it, so that's why we call it the 3' hydroxyl. And so that is how we're going to connect the next nucleotide. It's going to be connected. The sugar phosphate backbone is out here. These are the bases, right? And so it's going to be, you've got to bring in a C to hydrogen bond with this G. That this G tells us a C goes here. And so we bring that in and cut 2 phosphates off, and then basically connect it. And so now you have your growing end, the 3', you can see that here, 3' end, and you're going to have another 3' hydroxyl, so you can just continuously grow in that direction. Of course, something to consider is that if DNA is only synthesized 5' to 3', and it is only synthesized 5' to 3', then as you unwind the DNA, remember it's anti-parallel. So there's going to be one strand that's going to be perfect for you. You can just follow that, making your new DNA molecule, your newly synthesized DNA molecule is going to be constructed in a 5' to 3' fashion along a 3' to 5' template. But the other DNA molecule of the original template molecule that you just opened up is going to be, so we're at a replication fork. We have a 3' to 5' molecule, and we have a 5' to 3' molecule. That 5' to 3' molecule is oriented in the wrong way for us to simply just make DNA 5' to 3' off of it in the same direction as the other newly being synthesized molecule. So here's what I mean by that. So basically, if DNA is synthesized only 5' to 3', then as the DNA unwinds, one template is going to be correctly oriented, and the other is not. It's going to be reversed. And you can see that here. So imagine this is what we call a replication fork. It's called a replication fork because you have 1, 2, 3, 4 polynucleotides. So you can imagine those are the tines of a fork. And so basically the replication fork is unwinding, going from left to right across our screen. Obviously this is a static image, but you could imagine. And so as we're opening it up, you can see the top strand we write 5' to 3'. The bottom strand is 3' to 5'. And so if the bottom strand is 3' to 5', well then as this is opening up, going left to right, we can just synthesize DNA off of the bottom strand and just keep going. We just follow that. But the top strand is in the opposite direction. So we open it up to a certain point, then we start making some DNA, and we go the opposite direction. And then we're going to have to, this has to open up some more, so we could start here and then go this way to connect to this one. So one strand is perfectly oriented, the other one is reversed. And so when the template is exposed 5' to 3', that's the one that's reversed. So the template exposed 3' to 5' is perfectly oriented. The template exposed 5' to 3' is going to be the one that is reversed. Now, don't get me wrong, you could still use it. It's not that we can't use it, it's just that it won't be as smooth. So as the DNA unwinds, new DNA is synthesized continuously, smoothly along the lower 3' to 5' template, the one that's correctly oriented. That's called continuous synthesis or leading strand synthesis. You may have learned about these before in, like, say, Bio 161 or something like that, introductory bio, or even high school biology. And I hope that this is all familiar to you. I just have to go over it again because, of course, you know how when we went over the cell cycle, you may have seen that before, but even today we talked about how, oh, this is occurring during S phase of interphase. They all play together, and so it's important to have a good overall view when we start to get into the minor, the nuances of genetics. So continuous synthesis is the one that just follows along. You're synthesizing DNA and you're following the opening of the replication fork. So you can think of it as a zipper, right? As the zipper is being pulled and basically you're unzipping something, well, then you're just following that along continuously. So that's continuous synthesis. The new strand is called the leading strand if it's continuously synthesized, and that's why it's also called leading strand synthesis. So there's continuous synthesis, leading strand synthesis, and then newly DNA is synthesized along the other template strand, and that's the 5' to 3' one, but it's going in the opposite direction of unwinding. So you go in short bursts. And so then when you do that, you create a series of these fragments. And I'm sure many of you know what those fragments are called. Does anybody remember what those fragments are called? So if you're thinking Okazaki fragments, you are correct. And so only short lengths of DNA can be synthesized at a time in the discontinuous synthesis or the lagging strand synthesis. And as I said a moment ago, those lagging strand, the individual strands that are made, the individual fragments that are made along lagging strand synthesis is going to be made in a series of short segments, and those are going to be Okazaki fragments. And so they are going to basically show up, they're going to be synthesized opposite the direction of the replication fork. Replication fork, on the previous slide, it was going to the right. When you synthesize an Okazaki fragment, you'd be synthesizing that DNA on the screen from right to left. So here's an example. I like to use this example because this is leading strand synthesis. Again, I used 3-letter words just so that it's something that's easier than A, C, G, A, T, A, C, that sort of thing. But if it's leading strand synthesis, as this little sentence was basically laid out, you were reading it as it was being laid out. You didn't have to wait for it to finish. You knew you could just read it as it was showing up, and so it'd be the dog, the cat, or out. Alternatively, if it was lagging strand synthesis, you would have to wait, right? So we're waiting. There it is. The dog, the cat and the dog are out, but it started off as out, and then are out. What are out? Well, it turns out the dog are out. Well, there must be something else because that's not proper grammar. So and dog are out. Oh yeah, there's something coming. Cat and dog are out. The cat and dog are out, right? So that is lagging strand synthesis. These are individual little fragments, Okazaki fragments, and then they have to be connected. And so DNA ligase is going to come in and connect them. So Okazaki fragments of the lagging strand are joined by the enzyme DNA ligase. And then you have your DNA. And so here it all is put together with all the different things I may have mentioned in the past, or we'll mention them now. And so what we have is you're looking at a little bit more complex version of a replication fork. Replication fork is going from left to right again. We have the top strand, template strand, so that's 5' to 3'. We have the bottom strand, the bottom strand. That one is actually here. Let's see. I take it back. 5'. Oh, see, I lost it in there, but okay, 3'. Okay, so actually, it turns out that the top strand in this case because it's just the way that we're looking at the DNA, is the 5' to 3, sorry, I did it again, is the 3' to 5' template. So that's going to be our site of continuous strand synthesis. And the bottom strand, from our vantage point in this image, is the one that is going to be in the wrong direction. So you can see this is the 5' all the way to 3' direction. And so we have Okazaki. It even says old Okazaki fragments. So there's Okazaki fragments. And you can see here's an Okazaki fragment being made. And then here's an old one. And then they're going to be connected by DNA ligase. And so you can see there's DNA polymerase. We talked about helicases. So DNA gyrase relaxes supercoiling ahead of the replication fork. Helicase helps with that. There's also a primase that lays down RNA primers. And then you have single-stranded binding proteins. That's what SSB means. Single-stranded binding proteins help stabilize these single-stranded DNA molecules before another DNA molecule is made on them to make them double-stranded again. You've got a lot of different players. And so DNA replication takes place in 4 stages. Those 4 stages are the same 4 stages you run into all over the place. You know that there's going to be some sort of initiation in there, some sort of elongation, some sort of termination. Those are 3 that you find in all sorts of different stages of, say, transcription and translation. Well, it starts with initiation. DNA replication starts with initiation. In E. coli, so this is in prokaryotic cells, there's an initiator protein that recognizes and binds to the origin of replication, slightly unwinding the DNA. And then you can start to form, your replication bubble. Then you have unwinding. So unwinding is the initiation. It's the process of initiation lets helicase get started. Once helicase gets started, then it can fully unwind a short stretch of DNA. Single-stranded binding proteins, they're now little purple things, those come in, stabilize the strand of double-stranded, stabilize each strand of the double-stranded DNA molecule. So obviously they stabilize the single-stranded DNA. And then you have DNA gyrase, right? That relaxes supercoiling. I may have mentioned that on the previous slide. So I'll have a list for you here in a second, but you can see how this opens up and then continues to open up before anything has started. Before elongation can begin, where we actually start making some DNA, we have to lay down some primers. And so primase synthesizes short RNA primers to bind to these template strands, and that way you have a free 3' hydroxyl. You got to start building off of a 3' hydroxyl. And so now you can see primase is in there. Now we've got these little RNA primers, and so you can see DNA is being synthesized. So this is just going to follow the unwinding. So this is the leading strand. This is going to follow the unwinding. So that's the leading strand. There are 2 leading strands, right? Because we have 2 replication forks. They're kind of, in inverse of one another. So if this one's going left to right and 5' to 3' goes on top, then the bottom strand is going to give us our leading strand synthesis. If this replication fork is going, right to left, then the top strand is going to be our 3' to 5' template, and so we can make 5' to 3' off of that, and so that would be our leading strand synthesis. And then eventually we get enough space that we can get some Okazaki fragments being made. As this moves this way, you make an Okazaki fragment. This is now moved further over. Now you can make another Okazaki fragment. When this moves further over, we can make another Okazaki fragment, etc. And so elongation requires many different polymerases, right? We talked about all those different polymerases that had the lowercase Greek letters for eukaryotic DNA polymerases. And now what we have are the prokaryotic DNA polymerases. Those have Roman numerals. And so that's all I ask. I don't ask that you know. Here's that column again, that really brilliant column. So DNA polymerase, does it polymerase DNA? Yeah. And so basically what we have is I ask that you know that if you're looking at a DNA polymerase with a lowercase Greek letter, it's eukaryotic. If you're looking at one with a obviously uppercase Roman numeral, that's going to be prokaryotic. I'm sorry, I got sidetracked there because a fun fact just pumped into my head, and I don't know if most of you probably know this, but I'm going to say it anyway. So if it was a lowercase Roman numeral, what would that be? That's the final Jeopardy question for today. And so those are called Romanettes. So if this was like a little lowercase I, it'd be Romanette 1. If this was lowercase I, it'd be Romanette 2, etc. So if you knew that, then sorry to bother you. If you didn't know that, ta-da, you just learned a fun fact. All right, so now here's those lowercase Greek letters, right? So for the eukaryotic DNA polymerases. All right, so DNA polymerases work together to synthesize new DNA. So they're working all of these DNA polymerases, right? So you can see DNA polymerase that actually works to remove and replace primers. There's the one that actually elongates the DNA. There's DNA repair. They're not mutually exclusive. They're working together to make more DNA in the most high fidelity fashion possible. And so then lastly, the last step is going to be that NICs in the sugar phosphate backbone are going to be sealed by DNA ligase. So I said I had a list. So here's just, I'm sorry, before we get to the list, here's DNA ligase. So you can see, imagine two Okazaki fragments when they're adjacent to one another. They're basically going to be a little tiny, like they're hydrogen bonding with the bases just fine, but the sugar phosphate backbone is not fully sealed up. And so DNA ligase comes in and it will seal that up. So you got to replace your RNA primer. DNA polymerase does that. Then you have to seal the NIC, and that's where DNA ligase comes up. So the last step is going to be DNA ligase. And so finally, so when I say the last step, I meant the last step in the process, right? So termination usually occurs when replication forks meet. And so then everything falls off and DNA ligase can come in and seal up those NICs. So I should say, I guess I might be confusing here. So elongation, DNA ligase, then termination, where everything just dissociates, everything falls apart. There's the list. So here is what I would ask that you know. You need to know what an initiator protein is, helicase, single-stranded binding protein, gyrase, primase, polymerases in general, and ligase. So while there are quite a few players, the most important in prokaryotic DNA replication are these. And so make sure you're comfortable with the different DNA polymerases in terms of lowercase Greek letters for eukaryotic or Roman numerals for prokaryotic. Make sure you know what ligase is, what helicase does, what gyrase does, what single-stranded binding protein does. And if you know those, then you'll be well suited for any sort of test questions I could possibly ask you about DNA synthesis. And so don't forget that the high fidelity is achieved through proofreading and DNA repair. Those DNA polymerases are not mutually exclusive. Remember, they're working together. They're all working together. And so eukaryotic replication is similar to prokaryotic replication, but there are a couple differences we should mention, just so that we finish this out by making sure you know the differences between the two. So it differs in several aspects. So the biggest one is we have linear chromosomes and they're bigger. So eukaryotic replication must deal with a much larger genome and linear chromosomes, and we have nucleosomes. Remember DNA raptor and histone proteins? Those could be barriers. Those could be obstacles that the DNA polymerase is going to remember, it needs a smooth track, and if your train tracks wind around a boulder, right, that's going to put a damper on your travel. And so that's not certainly going to be a smooth track. So we have to deal with those nucleosomes as well. So initiation and replication are distinctly separated in eukaryotic DNA replication. That is to ensure timing, and it's also to help deal with all of these extra obstacles that we have. Origins, I mentioned before, are licensed by licensing factor. So remember when I was saying the linear chromosome, I showed you 3 origins of replication and they didn't all start at the same time? That's because licensing factor determines when you're going to start. And so it approves an origin for replication when all, when all actually are going to end at the same time. So it approves the origin for replication so that you can when you're completed with this process, that all the chromosomes will be completing the DNA replication point or process at the same time. And so many different DNA polymerases are employed, right? Alpha, beta, gamma, etc. And the most important are going to be the ones in the nucleus. Linear chromosomes mark a fundamental difference. So this is important now. We're getting to the biggest problem, which is the end replication problem. So linear chromosomes mark a fundamental difference between eukaryotic and prokaryotic replication. And because you have linear chromosomes, you are introducing another, a problem that simply does not exist in prokaryotic DNA replication, and that is the end replication problem. And so that's what this says. The most notable difference is the end replication problem. So let's talk about the end replication problem and what that is and what the solution is. All right, so let's start with circular prokaryotic chromosomes because they don't have an end replication problem. So they are ideal for replacing the RNA primer because the 3' hydroxyl is available. You just basically, if you started here, you just go all the way around, you come back, and you just go straight through that RNA primer. And so there is no end, and so therefore there is no end replication problem. And so you could see that replication around the circular chromosome provides a 3' hydroxyl group in front of the primer. Nucleotides can be added to the 3' hydroxyl group when the primer is replaced. So if we zoom in on that, you can see that here, right? So basically, it starts, here's your 5' RNA primer, 5' to 3' RNA primer, and then you have a 3' hydroxyl. So you start and you go all the way back around, and then you just go straight through, and you're all good. However, in linear DNA, this is also true for adjacent replicons, but not for the ends. So adjacent replicons, all right, well, this is going to get through there. This is going to get through. You'll be okay. But the ends pose a problem. And so you could see, for example, here's the RNA primer that was laid down so that we could start off of it and go this way. If you remove that RNA primer, then there's a chunk of this, part of this strand of the DNA molecule, the top strand, that you have to lay a primer down out here in order to replicate that portion. And so the theory would be, and this is not what happens, but this is the problem, would be that if you didn't have a way to remedy this, then you would lose a chunk of the ends of your chromosomes every time you replicated your DNA, those telomeres, right? Remember that? The ends of the chromosomes are telomeres. That's telomerase. Well, I'll get to the telomerase enzyme, but the telomeres are telomeric DNA at the end there. And you don't want to lose that. You don't want to lose your chromosomes. You don't want your chromosomes to be slowly, shortened every time you replicate your DNA, because, over time, even in your own lifetime, that's going to cause a devastating effect. But imagine just over, generational time, a couple generations, there's no chromosomes left. So that can't be. We have to have a way to synthesize the DNA here, or at least replicate, I should say, replicate the DNA or synthesize the DNA on this molecule. It's got to come in from this direction because we have to go 5' to 3'. And we need a primer out here so that we could start off of it and then use this template to lay down some DNA. Obviously, you can't have a primer out here because there's nothing for it to hydrogen bond to. So that's our problem. And we have a gap left by the removal of the primer. And the ends of these chromosomes are telomeres, and we don't want, remember they're associated with aging, we don't want telomeres to be degraded or lost, because once they go, then, it doesn't even matter. We're going to lose just chunks of our chromosomes, and we can't have that, simply can't have that. So telomerases are these enzymes I let slip a moment ago that are enzymes that extend the DNA. And they just add extra DNA. And it's just like filler, but it provides a template or a place for the primer to lay down. And so they are enzymes that extend the DNA, filling the gap left by removal of the RNA primer. Here's what that kind of looks like. So here's the gap left by the RNA primer. That's where the gap was. See the overhang, right? And we don't want to lose that DNA. Like this part we still have, but here we don't. And so that means that we're basically losing the information encoded in this little section of DNA, because eventually we're going to have a double-stranded DNA molecule and we're going to lose that, but we don't want to lose that. So telomerase is an enzyme that has an RNA template in it, and it basically sits down and it can extend the overhang. See that? New DNA, and when it extends the overhang, and it keeps doing it, just extends the overhang, eventually the overhang is going to be long enough that we do have somewhere to, remember how I said we can't lay a primer down out in space because we have nothing to bond to. Well, now we're not laying a primer down out in space. We have a primer, we have, DNA sequence that we can lay down a primer on, and then we can start synthesizing off of that towards our existing molecule, and that will synthesize the section right in here that we care about. So you can see that here. So now we've extended it tremendously. And once the telomerase leaves, then there's enough room to lay down a primer and then start synthesizing. And so now, like I said, a primer can be added and DNA can be synthesized to fill the gap, thus giving us a solution to the end replication problem. So when we replicate our chromosomes, we do not lose chunks of sequence, every single time. Well, at least we're not supposed to. We have a mechanism to replace that. But unfortunately, it can happen. But the natural mechanism is there. The telomerase enzymes are there in place in a healthy individual to prevent that. And so you can see here. So now you lay down a primer not shown, and then you start synthesizing DNA. And so now you make DNA that you don't care about, but you do keep the part that you do care about. Then eventually you lose the primer, the RNA primer. And so, if this gets degraded, that's okay, because we still have the section that we were worried about losing. We didn't lose it.